So it gives me great honor to invite to get back to Li-Fraumeni syndrome, Dr. Frebourg. So Thierry comes to us from France and is, I think, unequivocally one of the world leaders in this realm, and again, very honored to share the podium with him. Thierry.
So many thanks, Jeffrey. So first of all, I would like to thank my old friend, David Malkin. I would like to thank the LFS associations. It's a great honor and pleasure for me for two reasons. First, because the patients are daily motivation and source of inspiration. And of course, it's my great pleasure to meet all these colleagues working in the field since more than 20 years. So just to give the context of the work, our Department of Genetics is providing the P53 testing and the guidelines for Li-Fraumeni since more than 20 years for the entire countries. So we are in charge of the update of the guidelines of the education session, and we have now diffused that at the European level through the European Society of Human Genetics and European Society of Oncology. The idea is to try to diffuse the update data concerning the Li-Fraumeni syndrome. Coming back to the research part, our research laboratory is mostly involved in the functional interpretation because it has been very nicely highlighted by the previous speakers. I'm convinced as the medical geneticist, seeing patients every day, that the challenge is not anymore the mutation detection, but is interpretation of the mutation. So definitely we need a certain level of expertise. So just to start with that, as you know, and many thanks for my colleagues who have cited this work, we published in last summer in the Journal of Clinical Oncology this revisiting, it's a new update of the ChIP-seq criteria, and in fact we have been able to identify now more than 500, but we try to give an update of the clinical presentation starting from more than 415 P53 mutation carriers, among which we had 322 affected mutation carriers who developed a total of more than 550 tumors. And here's a final version of what we can call the ChIP-seq criteria. First, you have the classical familial presentation characterized by two relatives presenting with the classical tumors observed in LFS. The second box of presentation corresponds to multiple primary tumors because we know that we have an increased risk of multiple primary cancer. The third box corresponds to the development of rare tumors, and we have included this canonical, very suggestive of LFS corresponding to adrenocortical carcinoma, choroid plexus tumors, and some subtype of rhabdomyosarcoma. And finally, and I completely agree, the question of early onset breast cancer as defined before 31 years of age. Just to highlight how it's very important in a clinical practice, if we are looking at the mutation detection rates of patients fulfilling only these criteria without any additional criteria for the Li-Fraumeni syndrome, we had a mutation detection rate in childhood adrenocortical carcinoma of 45%. For choroid plexus carcinoma, we had a mutation detection rate of 42%. And when we look at the early breast cancer, that was the early breast cancer before 31 years of age, we had a mutation detection rate of 6%. So we fully agree with Garrett, with colleagues from US, that if you take that as the only criteria, without any additional criteria for LFS, the mutation detection rate for P53 mutations in very early breast cancer is 6%. We can consider that this number is stabilized and is definitely the first genes for early breast cancer before 31 years of age, explaining why P53 is becoming more and more important into the field. Now, the second message that we deliver, also we have to be very careful, and of course the class of the mutation is not the only explanation of the diversity of the phenotypic presentations, we had strong evidence for what we can call a clinical gradient of germline TP53 mutations. So we have three levels. We have, of course, as it has been mentioned by previous speakers, we have what we can call the more severe mutations characterized by high penetrance that correspond to the missense mutations with an established dominant negative activity as demonstrated by functional assay, usually performed in yeast or human cells. And you have here some example affecting codon 248 or 282. They are usually associated with early tumor onset, and they represent the predominant germline alteration in childhood cancer, except ACC that correspond to a very specific item. Now, if you are looking at the bottom of the scale, what you have are the less severe alterations characterized by a lower penetrance. These mutations are what we call loss of function mutations. So they are going to destroy the protein. They correspond to nonsense mutations, to frame shift mutations or genomic rearrangements, and usually they are classically associated with later tumor onset, and they are predominantly detected in pedigree with late tumor cancer. And of course, we have this specific class that we can call, also it's a very reductive, the non-dominant negative missense mutations, and we have the famous example of the Brazilian mutations, the ARG337IS, and we have the equivalent in France. We have identified as the predominant mutation associated with ACC, a similar mutation affecting codon 158, ARG158 histidine, and usually you have a typical presentation, ACC, only ACC or ACC, and late onset cancer, mostly breast cancer occurring in female. So now what I would like to do is to summarize all the data that we have accumulated so far to establish a molecular rationale to this clinical gradient of severity. So as you know, we had the surprise to identify this particular mutation. If you look at this pedigree, 10 years ago, have we been able to indicate that this mutation, that this family corresponds to a real Li-Fraumeni syndrome, because we had here only very late onset cancer without any childhood. If you are looking here, for instance, at the age of the tumors in the index cases or within the family, and we identify here a genomic deletion, so it's a pure null mutations. If you are looking at another pedigrees, we have the same story. You have here a male who was free of cancer until 40 years of age, and suddenly, after 41, he developed subsequently 4 other tumors. And we have the surprise to identify here a stop mutation, which is predicted also to result only in a null mutations. And finally, as another example, who gave us an highlight of that, we had the example of these mutations corresponding to a splicing mutations associated also with late onset cancer and probably corresponding to a late onset cancer. It's the reason why I think, and we had this discussion during the dinner last night, that in order to integrate the heterogeneity of the phenotypic presentation of LFS, it would be a good idea not to switch and to change the title of the syndrome, but to extend and to expand that from LFS to P53-related cancer, according to the model of cystic fibrosis. Cystic fibrosis is due to biallelic mutation of CFTR, but we know now that we have a CFTR-related disorders, and I think it would be a good idea to move, because for patients, it's something to say you have a Li-Fraumeni syndrome, and it's not the same thing to have a null mutation associated with late onset cancer and to have a dominant negative mutations affecting the children in your family. So that's something that we can discuss all together, and especially with the professional and also with the associations of patients. So to come back to the molecular basis of what we have observed here, so we took advantage of the key role of P53. So as many previous speakers highlighted, we know since David Lane's work in the late 90s that the key function of P53 protein is acting as soon as you have DNA damage occurring directly or indirectly, for instance, after oncogene activations. And as soon as you're going to have DNA damage, the P53 level will increase, and P53 will act as a very powerful and universal transcriptional activator, able to upregulate the transcription of genes either involved in DNA repair, in apoptosis, but also in cell cycle. So in order to explore that, we decided a couple of years ago to develop a new functional assay, but not in yeast, what we had done with colleagues 10 years ago, but definitely in the cells of patients. So I had presented that previously. So the assay that we had developed is the following, is based on the induction of DNA damage. So first, you are starting from the cell of the patients. On a routine basis, we are doing that either on fresh lymphocyte or lymphocytes that have been previously immortalized by EBV. What you are going to do is you are going to induce very briefly a DNA damage, and we are using different types of drugs. Usually we are using doxorubicin able to break the DNA. And after that, we are doing a transcriptome analysis, and we are doing a simple comparative transcriptome analysis between exposed cells and unexposed cells. So it's a comparative analysis, but you are using the same cells of the patients, you are working in the same genetic context. And what we had published previously was the following. If you do that in normal cells, so in a lymphocyte with a wild-type phenotype, with a wild-type genotype, we have here the number of probes corresponding to number of genes which are upregulated after the DNA damage. And you have here the result that we obtain with lymphocyte derived from LFS patients with pure null mutations, such as frame shift mutations or complete deletion, for instance. As you can see, you are here, you are working the lymphocyte, you are working the lymphocyte, so you have only an heterozygote mutations. And you see, of course, a slight decrease of the number of probes. And if you are looking per genes, you see a slight decrease of induction. But now, if you do the same experiment with lymphocyte, not transformed cells, but lymphocyte from LFS patient with a dominant negative mutation, as you can see, you have a huge decrease in the number of probes, therefore in the number of upregulated target genes. And if we look very carefully at the level of induction, we have a decrease of the level of induction. So you have here the demonstrations that these severe mutations associated with a severe clinical phenotype, these dominant negative mutations are able to drastically alter the transcriptional activity of P53 when DNA damage occurs. So we have here what we can call, as scientists and physicians, an endophenotype, before even the occurrence of the cancer. So since the publications of that, we decided to develop a simple assay in order to integrate that on a routine basis. So the idea is to do the same. You have the lymphocyte, either fresh lymphocyte or the EBV immortalized lymphocyte, and you are going to expose that to doxorubicin. Instead of performing transcriptome analysis that you cannot do for every patient, what we decided to do was to perform a comparative targeted analysis. So we selected from the transcriptome data 6 different genes which are being to be upregulated by the induction of DNA damages, and we designed a simple assay which is called a reverse transcription QMPSF assay. So what you are looking here, you have different peaks, and you have here, for instance, in blue, you have the different peaks of the different genes that we have selected. You have here what we can call control genes, so they are not going to be changed by the DNA damage. And here you have what you are going to expect if you have an induction. And what you are just going to do is you are going to superimpose the profile of unexposed cells with the profile of exposed cells, and you can see, I think, the level of induction. And we arbitrarily define what we call a functionalist score on the basis of the mean of the induction level of the 6 P53 target genes that we selected from transcriptome analysis. So now if you look at the results, so we are working in the lymphocytes. So if you are taking, for instance, the result from normal lymphocyte with the wild type P53 genotype, we had a mean of functionalist score of 14. When we did the same experiment with null mutation corresponding here to a complete deletion, we had a decrease, but it was like a 50%, 7.2. But when we did the same experiment with dominant negative, as you can see, we had a huge decrease of the score. So you have definitely here a clear demonstration of a phenotype of an heterozygous mutations in the genetic context of the patients. So the next slide shows you how the assay is reproducible. So we have done many assays and corresponding to different families or different members among the same family, and you can see that the functionalist score is very similar. So we were very confident with the assay, and we decided to extend that to many families that we had analyzed. So on the next slide, what you can see is using this routine functional assay, you have here the presentation of the functional score that we had estimated. So we have extended that now to 14 null mutations predicted to destroy only the function of the protein. We had extended that to 16 dominant negative mutations. And as you can see, if you have the functional score of different controls, here of null mutations, of the dominant negative missense mutations, and you can classify these mutations in the two different mutations into different classes. Then we decided to analyze other mutations that had not been characterized so far. So we took a range of more than 26 missense mutations without established dominant negative. So this mutation has been considered as non-dominant negative, or either they had not been analyzed so far in the different assays performed in cultural cells or in yeast. So you have here the list of the mutations corresponding to 26 missense, to 26 different French families that we have analyzed so far. So some of them are located in the DNA binding domain, in the transcriptional activating domain, in the hinge region, or in the tetramerization domain. And what you can observe is you have a dispersion of the functional scores indicating that these mutations are very heterogeneous. So we decide to go a little bit further in details, and when we did that, first we start by these mutations which were located into the hinge domain. And it's very interesting to note that if you are looking at the phenotype, so two of the mutations have been identified in late onset breast cancer, and these mutations have been identified in, and according to me and according to the previous speaker, irrelevant phenotype. It's probably an incidental finding because we have no evidence of the involvement of LFS in teratocarcinoma. And as what was interesting is that in the Cato work performed, especially in yeast, those mutations have been classified as super wild type. And as you can see, we had a functional score very close to the wild type. So we have no evidence that these mutations were really deleterious mutations. Of course, we are just looking at the transcriptional activity, but so far we have no evidence of real deleterious mutations. Now, when we looked at the famous Brazilian mutations and the similar French mutations, what we observed, we had a functionally score which was very close to the null mutation. So the result we got fully agree with Professor Zambetti's presentation, indicating that we are dealing with an hypomorphic mutations, not a very penetrant mutations. And finally, when we analyze the other mutations that had been not classified so far, we had a drastic alteration. So I think that this assay, among other types, of correlation will be helpful in order to classify these mutations we are going to be identified mostly by now panel genes. So now to finish and to go a little bit in details, since we had the demonstration of a drastic alteration of the transcriptional activity, we decided to go a little bit further. And we decided to look at the impact at the genome level of these deleterious mutations. And for that, we use a panel of what we can call the ChIP-seq experiment, which correspond to the chromatin immunoprecipitation and deep sequencing. So the idea is to see how a mutation at the heterologous status can impact the binding at the DNA through the genome. So to do that, we did the following experiment. First, this required many steps of optimizations. So the idea is you take the lymphocyte, you activate p53 pathway by the classical genetic stress using doxorubicin. You have here an examples. After that, you are going to perform a self-fixation analysis in order to bind p53 to the corresponding target. After that, you are going to cut the chromatin by ultrasonications. You have to optimize the condition. And after that, we immunoprecipitate the bound chromatin and we perform deep sequencing. So we use here a high throughput and an assay. You have here the number of reads. And I will go now to that because it was very important to optimize the bioinformatic analysis. So it's a little bit tricky, but you have here the pipeline when you are doing that. So you have to sequence all the bound fragment on which p53 has been bound, and you have to find the peaks. You have to filter the peaks in order to be sure that you are specific. After that, you have to center the peaks. You have to find the sequence, and you have to perform an annotations. So basically, the results are the following. I'm going to just to give you an highlight of what we can go to see and to convince you how accurate is this type of analysis. Here, for instance, you have a table with some very classical genes that we found, like PUMA or P21. And here you have the location of the bound region. And as you can see, we just had just upstream the transcription site. So definitely we were able to locate it where p53 was able to bind. And here you have two parameters which ensure the quality of the experiment. The peak score corresponding to the read numbers, the number of reads obtained by NGS. And here what we can call the focus ratio. You should get the same number in both strands. Now, what was very interesting is that when we did the experiment in wild type 53, we observed a huge number on 2 replicates. So we obtained more than 700 bound regions. And remarkably, when we analyze that, 92% of the detect peaks contain the famous 53 binding motif, indicating definitely that the experience was okay and we had a good specificity of the ChIP-seq experiment. Now, in order to check that, we perform biological analysis, we derive how functional assay develop in yeast, and as soon as we have the fragments not identified by previous publication, we clone it in a promoter with a defective promoter and in a plasmid and containing the add2 open grading frame. And to make a long story short, if 53 is able to bind to this region, you are going to activate the defective promoter, you are going to increase the expression of add2, and you are going to switch the color of the colony from red to white. And you have here a biological demonstration of the binding of 53 to the region. For instance, here, if you do the experiment, you can see that, for instance, this unknown gene corresponds to a new, for instance, target P53 genes. So it's probably one of the nicest slides that we obtain. Now, we did that specifically for some genes. So you have here the basic results that you can visualize using this, for instance, using this software. Here, for instance, you see the number of reads that we obtain in the wild type configurations. So you can see definitely, and it's very nice, you have here the binding of 53 just upstream the transcription site of the the canonical gene 53, one of the most famous genes. If you do that now in lymphocyte from LFS patients harboring a missense mutation, a dominant mutations, what you are going to observe is the following. You can see very easily that you have more than 50% of decrease of the reads, indicating that definitely you have the visualization of what is a dominant negative mutations, highlighting the fact that you are going to impact at the genome scale the binding of 53 to the corresponding target genes. And if you are looking now not only at specific targets but all the targets, you have the results that we obtain here for the wild type, here for the LFS lymphocyte, and as you can see, we had a huge reductions through the genome, at the genome scale, of the different target. So you have an alteration at the genome scale of the binding of p53, and if you reduce the size to region close to the transcription site, as you can notice, we had a huge number. So now we had a massive reduction of 53 DNA binding, but we had no evidence of an aberrant binding by the mutant. I say that because, as you know, there is always this discussion between loss and gain in function, and as you can see, we do not have evidence of an up-regulation. So just to finish on that, there was a very nice paper published in the last fall using the same techniques not in immortalized lymphocyte, but in cell lines. And what the authors from the Pennsylvania University obtained is that they observe that when you have missense mutations, as Carol Price clearly highlighted, you can have an upregulation. We did not find that. We checked the result. We went back to our transcriptome analysis. We did not have the same result. So I don't say that the results are not okay, but we are here working before the occurrence of cancer, and our colleagues from the Pennsylvania University had done that at the cancerous levels. So just to finish, because I'm a bit out of time, I think that we can give now an integration of all the data. We know that definitely the basis of Li-Fraumeni is a loss of functions. We have evidence for that. We have different types of mutations, but we have especially loss of function mutations associated with Li-Fraumeni syndrome or 53-related cancer. We had previously demonstrated that using the yeast assay, you are able to destroy the functions, so definitely the Li-Fraumeni is a loss of functions. Now we have additional data demonstrating that definitely we have a drastic alteration of this type of mutation corresponding to the dominant negative mutations. This has been due to a drastic alteration of the binding demonstrated by the ChIP-seq experiment, and this explains probably the clinical gradient and the clinical severity of these mutations, explaining why they are predominant and they are associated with severe presentations. And just finally, and that will be my last slide, I think that we provide evidence of the molecular basis of the clinical gradient of germline TP53 mutations. And as a clinician, I think that we should consider, maybe, it's a question of discussion, that it would be a good idea to stratify the clinical management according to the type of this mutation, because you should have in mind the balance between the psychological impact of delivering a p53 result and the real medical benefit. And what we have suggested to consider, at least in France, as a pilot study, is that it could be maybe a good idea to reserve the presymptomatic testing in children, and the protocol developed by our colleagues from Toronto based on annual MRI in patients harboring those dominant negative mutations. And in contrast, in families with loss of function mutation characterized by late onset, to consider presymptomatic testing only in adults, with the idea, as Garrett showed very nicely, to perform annual breast MRI from 20 years of age, and of course for the intermediate class corresponding to the famous Brazilian mutation and the other, to do presymptomatic testing, but with the idea to perform a selective detection of ACC and also and annual detection of the breast cancer. So I will finish here. I would like to thank all the collaborators that represent 20 years of work, of collection, thanks to my colleagues Gaël Boujard, functional essays performed by Jean-Michel Flamand, and all the colleagues here. And I am very happy also to share all the results. Most of them are unpublished so far. Thank you very much.